This is the Clostridium and other anaerobic rods tutorial presented by the SchrDbb. We reference Lippincott Illustrated Review of Microbiology by Cynthia Cornilesson, Bruce Fisher, and Richard Harvey. So here we're going to start with the classification of the rods, these other rods here. So we start by free living organism. We need to know that what the Clostridium species are under the gram-positive rods. So we have all seen other gram-positive rods. The gram-positive rods you have been seen already. You have seen Bacillus, you have seen Corynebacterium, you have seen Listeria. So the Clostridium species is under the gram-positive rods. Is it clear? Now the other gram-negative rods that we are going to see are more of anaerobic. So the other gram-negative anaerobic rods are going to be the Bacterioid species and we have the Bacterioid species here. Example is Bacterioid fragilis. Is it clear? And we have also Fusobacterium species. Is it clear? So those are the two main gram-negative anaerobic rods. Is it clear? Now for the gram-positive rods are mostly the Clostridium species, and under the Clostridium species you have Clostridium botulinum, you have Clostridium difficile, you have Clostridium perfringens, and we have Clostridium tetani. So Clostridium difficile is the one that causes the pseudomembranous colitis. Is it clear? Clostridium botulinum is one that causes food poisoning with botulinism, where you have paralysis of the muscle. Clostridium tetani is the one that causes tetanus, where you have paralysis of muscles, where you have tetanism of muscle when you have a wound infection caused by Clostridium tetani. Is it clear? And then the last one is Clostridium perfringens. This one is due to is going to be causing the gas gangrene. So it's a form of a unilateral swelling where you have gas underneath the skin. Is it clear? So these are the toxic effect of the reactive oxygen and intermediate on the anaerobic bacteria. So you need to know that anaerobic bacteria don't like oxygen at all. At least if you're a facultative anaerobe, you are going to thrive at least with oxygen. But if you're an anaerobic bacteria, you don't like oxygen at all. Is it clear? So this is why there are problems when there is oxygen. So this is a case where, let's say that there is oxygen at the level of the bacteria. We see that the oxygen, so oxygen can bind with an electron to produce superoxide. Oxygen can bind with 2 electrons to produce the hydrogen peroxide, and oxygen can bind with 3 electrons to produce the hydroxyl radical. So these are the families of reactive oxygen species. Is it clear? These are reactive agent or the reactive oxygen species. Intermediate are formed by partial reduction of molecular oxygen. They are highly reactive and can cause serious chemical damage to the DNA, protein, and ultimately leading to death of bacteria which are anaerobic. So anaerobic bacteria do not like the presence of oxygen at all. So they are going to live in places where there is no oxygen at all. Is it clear? So because these superoxide, hydrogen peroxide, and hydroxyl radicals which are present which are formed from oxygen are going to destroy this bacteria. They don't have any catalyst enzyme, any form of enzyme that's going to reduce all this. Is it clear? An oxidase enzyme to reduce all that. So they are very afraid of oxygen. Now these are different cases. So the first tutorial we're going to, the first microclostridium we're going to see is Clostridium perfringens. So what is the virulence factor of Clostridium perfringens? First thing is that Clostridium perfringens produces exotoxins, and examples of the exotoxins is alpha toxin. Alpha toxin is going to result in damage of the mammalian cell, resulting to the release of the lysosomal enzyme. So and that lysosomal enzyme is going to destroy the surrounding tissue. Also, the alpha toxin is going to cause the lysis of the erythrocyte, the leukocyte, and the platelets. Now, the next cause of Clostridium perfringens is also going to be the next virulence factor is going to be enterotoxins. So the toxins, these enterotoxins, are capable of entering the enterocytes and cause the release of the CAMP, and that CAMP now makes the proteins and the water to be released in the intestinal membrane. So the toxin alter the membrane, leading to less fluid and intracellular proteins to enter the enterocyte. Now the other degrading enzymes which are present in Clostridium perfringens, we have the DNase. DNase can split up the DNA, so it degrades the viscous DNA in necrotizing tissue or exudate, aiding the spread of the infection. So when you use these DNase, as I've already said in the tutorial part, in the past section, there are certain enzymes which can split up the DNA of the dirt Clostridium perfringens, or the death bacterium. Those enzymes are called DNases, and those DNases are capable of splitting the DNA that contain genetic information to counteract the immune system of the host. Is it clear? So when splitting the genetic information into many parts like that, it can easily spread that genetic material all through the body, such that the bacteria that have not yet overcome the immune system knows the information to overcome that immune system. Now the next is, it also produces the enzyme called hyaluronidase, and that hyaluronidase is used to break down hyaluronic acid, which is present at the level of most connective tissue, to increase the spread of the disease. Now this is an example of disease caused by Clostridium perfringens, which is called gas gangrene. So gas gangrene, or 5 cm superficial laceration on the left and the cubital depression. Is it clear? So this is following surgical debridement. You have this, so the patient has the gas gangrene. So Clostridium perfringens, this is the cultural measure of Clostridium perfringens, where you see colonies on the blood agar. Is it clear? You see a double zone of hemolysis. Is it clear? So this is the colony on blood agar. Now this is the gram stain of Clostridium perfringens. So you see that it is more, you can see purplish color of the rods. So that's why you see that Clostridium is a gram positive rod. Is it clear? As you can visualize, you can see purplish color of the Clostridium perfringens, just to tell you that Clostridium perfringens are like other Clostridium, are gram positive rods. So now we are going to move now to the next Clostridium species, which is Clostridium botulinum. So for Clostridium botulinum, the main pathophysiology of the disease is via the botulinum toxin, and the disease is called botulinism. So here what happened is that normally you need to know that without the presence of botulinus, when you have action potential at the level of at the end of the action, at the end of a nerve fiber, you have what is called a synapse. Is it clear? At the end of a nerve fiber, you have what is called a synapse, and that synapse is made up of the presynaptic membrane and the postsynaptic membrane. In this case, the postsynaptic membrane is the muscle membrane. Is it clear? The sarcoplasmic membrane. Now, at the end of a nerve, that's a nerve terminal, you need to know that what action potential is going to be converted from an electrical stimulus to a chemical stimulus. So what happens is that when the action potential is going to be transmitted, calcium ion is going to enter the cells. Is it clear? When calcium ion enter the cell, it's going to bind with the vesicles containing neurotransmitters, and in this case, the neurotransmitter that activates the muscles is for somatic sensation, for somatic movement or motor movement, is going to be acetylcholine neurotransmitters. Is it clear? So it binds with acetylcholine vesicles. So calcium binds with acetylcholine vesicles and binds with the presynaptic membrane to result to the release of acetylcholine. Now acetylcholine now is going to bind to acetylcholine receptors at the postsynaptic membrane, in this case the sarcoplasmic membrane, to produce a muscle contraction. Is it clear? So that is the normal action potential. So that's the normal action potential pattern. Now, in case where you have ingested now a food substance with a botulinum toxin, so you are going to have the botulinum toxin that is going to enter the enterocyte and going to be released to the bloodstream. Is it clear? So the botulinum toxin consists of two main chains. It consists of the light chain and consists of the heavy chain that can be transmitted in blood. So it's going to enter at the level of the nerve terminal, at the level of the synapse, at the level of the presynaptic membrane. So when it enters there, it's going to impede the binding of calcium and the acetylcholine. Is it clear? The acetylcholine neurotransmitter. So when it impedes that binding, so when the patient wants to, let's say that the patient wants to move, to do voluntary movement activity. So let's say that you want to move your hand to take your pen. So in order to move your hand to take your pen, normally acetylcholine is supposed to be released via this mechanism, and acetylcholine is going to be released to your muscles for the activation for muscle contraction. But when you have botulinism, you are going to be paralyzed. Why? Because this botulinum toxin containing the light and the heavy chain are going to inhibit the binding of the calcium and the acetylcholine vesicles together. So no acetylcholine is going to be released, no muscle contraction. You are going to have a flaccid paralysis. Is it clear? So that is for Clostridium botulinum. Now the next mechanism is for tetanus. So how does tetanus occur? So tetanus, that was a normal pathophysiology. In case of tetanus, tetanus toxin is going to block the release of inhibitory neurotransmitters such as the gamma aminobutyric acid and the glycine. Is it clear? For that case, for a case of botulinum toxin, it's going to inhibit the binding of the presynaptic vesicles with the synaptic membrane. Is it clear? So it impedes the release of neurotransmitter into the synaptic clefts. So now, in this case here, in tetanus toxin now block the release of inhibitory neurotransmitters. You need to know that there are certain neurotransmitters which are helping to inhibit the activity of the brain, while other neurotransmitters help to excite the activity of the brain. So examples of neurotransmitters that excite activity of the brain is glutamate or glutamic acid. An example of neurotransmitter that inhibit the activity of the brain is gamma aminobutyric acid, that's GABA, and also glycine. So this tetanus toxin inhibits this they block the release of this inhibitory neurotransmitter, resulting to excessive excitation at the level of the muscles. So what happens is, do you have these inhibitory neurotransmitters here? Now in presence of the toxins are going to enter. Now in presence of the toxins, it inhibits the release of the inhibitory neurotransmitters. So when there is no inhibitory neurotransmitters, the postsynaptic neurons are always going to be activated. Since the postsynaptic neurons are always going to be activated, it's now going to produce what is called tetanus. Tetanus means that what? It means that excessive action potential is going to be transmitted at the level of the muscle, and the muscle is going to be infinitely contracting and never going to stop. Is it clear? The muscle is going to infinitely contract because there is excessive postsynaptic neuron action potential. So the muscle is going to infinitely contract because there is no inhibition of those action potential. Is it clear? So they're going to produce tetanus. Now the question that you have to know is that tetanus does not occur in cardiac muscles. Tetanus also do not occur also in smooth muscles. Is it clear? Tetanus do not occur in cardiac muscle and smooth muscles. The question I want to ask you is why? Why is that tetanus only occur in cases of skeletal muscles and not in cardiac muscles nor smooth muscles? So you need to know that in patients that have tetanus, the patient are going to show things like jaw, so they are going to show trismus, that is jaw clenching of the jaws. They are going to show opisthotonous position. They are going to show different muscle contraction abilities. Is it clear? But now you need to know that the heart of that patient is going to be contracting normally. It's not going to be contracting in this pattern. There are two reasons why the cardiac muscle is not going to have this tetanus. The first reason is because the cardiac muscle has a pacemaker, and it does not depend on the activity of the central nervous system that the cardiac muscle is contracting. That's the first reason why cardiac muscles do not have tetanus. The second reason why the cardiac muscles are not attaining a state of infinite contraction, that is tetany, is because the action potential of the cardiac muscle is equal to the contraction time of the cardiac muscle. Is it clear? So if you have an infinite number of action potential, it is just going to be the same time at which the heart muscle contracts. So the heart muscle is not going to permanently contract in a tetanic pattern. Is it clear? So if you're not understood, it's better to go on our tutorial that we did initially on physiology, that is cardiovascular system physiology. Now, the next thing to visualize here is the Clostridium tetani. So these are the Clostridium tetani spores that you can see here on the photo micrograph. Now, these are the frequently used medications. You can use ampicillin, you can use amoxicillin, you can use cephalosporin, you can use clindamycin. Now, occasionally associated, you can associate penicillins other than ampicillin, sulfonamides, erythromycins, and trimethoprim with quinones are occasionally used. Rarely used are these other drugs. Now, what are so for Clostridium species, this is generally the general characteristics, the summary. So it is large blunt-ended rods that produce endospores, and this is the second microorganism that produces endospore after Bacillus anthracis. Is it clear? So it produces endospore. Most species are motile. Most Clostridium species are motile. They are obligate anaerobes. Is it clear? They are obligate anaerobes, but they are gram positive, and they are they culture anaerobically on blood agar. So this is their culture on blood agar, Clostridium, and an example is Clostridium perfringens. And this is the gram stain. You see that it is purplish color, and you may also see endospores in them. Is it clear? Just to tell you that this is Clostridium species. Now the next is, this is an example of Clostridium perfringens, is an example of Clostridium tetani. So what are the causes of the disease in Clostridium perfringens? You have myonecrosis, you have food poisoning, Clostridium perfringens, you have anaerobic cellulitis, you have necrotic enteritis, and you have Clostridial endometritis. To treat Clostridium perfringens, you just use your penicillin or you use your clindamycin. Now the next is going to be Clostridium tetani. So Clostridium tetani is going to cause tetanus, and to treat Clostridium tetani, you have to make sure that you use anticonvulsant, anti-tetanic drugs. So you have to use muscle relaxant drugs. You have to use anti-tetanic drugs like muscle relaxant drugs. Is it clear? And then the other antibiotics that you use penicillin G, you use metronidazole, or you use doxycycline in the second line. Is it clear? Those are the drugs you use for tetanus. Now, so this is how myonecrosis resembles. So there is necrosis of the muscle in the case of gas gangrene, and this is how tetanus resembles the patient in opitotonous position. So this is not a clear typical opitotonous position, but you can see extension of the level of the spine here. Normally in patients with this, the calf muscle is not like this, but it is the leg is also extended. So the next one here, this next here is Clostridium difficile. So in Clostridium difficile, is going to produce pseudomembranous colitis, and the main drug used in Clostridium difficile first line is metronidazole. So metronidazole is the first line drug in Clostridium difficile. Second line drug is vancomycin. Now the next is, so you can use pseudomembranous colitis. So this is a diagram showing pseudomembranous colitis, how the colon is going to resemble when you have an infection of Clostridium difficile. And Clostridium difficile is one of the most common hospital-acquired infection when you use excessively antibiotics. So when you use excessive antibiotics, it's going to kill your normal enteral normal flora. Is it clear? When you kill your normal enteral flora, it's going to cause a Clostridium difficile infection of the colon. Now this is a Clostridium botulinum causing botulinism. So you see how this patient is paralyzed. Usually there is no antibiotic that is given in patient with botulinism. Is it clear? Usually no antibiotic is given to them because it's caused by the botulinum toxin. So you are going to try to prevent the child from dying by so it's just a care to make sure that it is supportive to the patient. So you have to give oxygen to the patient because of the respiratory muscle do not function. They are paralyzed in case of botulinism. So the next anaerobic microorganism, we said they are Bacterioids microorganism, and we said that oxygen can easily kill those organisms. So anaerobic bacterium, we have facultative aerobic bacterium. For anaerobic bacterium, you see when oxygen comes in facultative aerobic bacterium, they convert that oxygen to produce water, and there is growth of the facultative aerobic bacterium inside the media. But in the case of anaerobic, you see oxygen comes, there is beginning of growth of anaerobic bacteria. Here they are only going to convert oxygen to produce water. For anaerobic bacteria, anaerobic infection often involve co-infection with facultative aerobic. Is it clear? So if you want, usually when there is an infection with anaerobic microorganism, there is a co-infection with an aerobic microorganism. Why? Because the aerobic microorganism are going to use the oxygen in the milieu to convert it to water, such that the anaerobic microorganism are going to grow. So an example of a co-infection of both anaerobic and aerobic microorganism is going to be a case of a necrotizing fasciitis, where the aerobic microorganism are going to be the streptococci, and the facultative aerobic microorganism are going to be streptococci, and then the anaerobic microorganism are going to be the bacteria. It's a polymicrobial effect. Another case can also be in a case of an abscesses formation. Is it clear? So, or even the Fournier gangrene. So from here we have visualized all the different things involved in microorganisms. In cases of other gram negative, other gram positive and gram negative anaerobic rods. So say thanks for your kind attention. Don't forget to like and subscribe for our Science Video Maker.